Good morning. Welcome. My name is Alfonso Valencia, and it's for me a pleasure and an honor to welcome you to the European Conference on Computational Biology, ECCB 2020. This is the 19th European Conference in Computational Biology. This time is organized by the Barcelona Supercomputing Center, the Spanish Bioinformatics Institute, and with the collaboration of the International Society for Computational Biology, ISCB, and the European Infrastructure for Computational Biology and Bioinformatics, ECCB. As you realize by now, we are not in the wonderful city of Sitges with parties, sun, and beaches, and instead of that, we are organizing this conference virtually. Obviously, we are missing a number of things. At the same time, there is an interesting new possibilities using the technology. In this case, we have made a more open conference. We have reorganized the program in a way that this is accessible to bioinformaticians around the world. Indeed, the participation in number of submitted papers and speakers from other countries has been larger than in previous conferences. In particular, we have adapted the program to allow the participation from other regions with a special session dedicated to bioinformatics in Latin America, for example. We have also made a special effort in terms of equality and gender issues, organizing a session together with the program of bioinformatics for women with different activities, including special sessions and a keynote speaker in the last day of the conference. This huge effort is possible thanks to my co-organizer, Salvador Capella, that will introduce to us the program of these days later, and also to the number of people that have been working in the committees, including the members of the steering committee, Arcadi Navarro, Fatima Sharur, Christine Orengo. And also, of course, the helps of the local organizing committee, Eva, Edurne, Jen, Ioannis, and Alberto. The collaboration of area chairs for the different topics, 24 area chairs and 100 referees that have helped us to evaluate the papers that will be published in the special issue of the conference in bioinformatics, the journal. And of course, the sponsors, including the ABI GmbL, Goblet, the Spanish Supercomputing Network, St. Jude's Children's Hospital, Bioinformatics for Women, GENES, the Journal of NDPI, Cambridge University Press, and the pharmaceutical company Grifols. So thanks to them, and welcome to all of you. We hope to have a very interactive conference. And now let Salva introduce the organization of the conference for the next 2 days. Thank you and welcome again. Salva.
Thank you, Alfonso. Welcome everyone from far and from near. ECCB 2020 has totally changed its traditional format to adapt to a virtual format, as Alfonso was mentioning before. It has been a challenging task, but also an exciting opportunity to propose a new structure for the conference and the satellite events. We have already celebrated 2 major satellite events, the New Trends in Bioinformatics by ECCB, which brought together during a week, last week, 19 tutorials and workshops in compacted session of 3 hours to facilitate attendance by everyone. We had 7 limited capacity sessions to favor interactions among participants and so on. And the rate of attendance was around 60 to 90%. And the rest of the session, 12 of them, had in many cases over 100 participants, with some of them even reaching 200 participants. Topic-wise, those sessions cover a wide variety of topics, including the latest development of machine learning, the use of PubBio, the long-read sequencing technology for RNA-seq analysis, the use of HPC for cellular processes simulations, and so on and so forth. We also welcome the 5th annual edition of the European Bioinformatics Core Facility community as part of the New Trends in Bioinformatics, and workshop by our colleagues from ECCB, our strategic sponsor, including one with the Global Alliance for Genomics and Health. The second satellite event that we already had yesterday was the 5th European Student Council Symposium, which took, we have more than 100 participants and has 2 keynote by Patrick Alloy, member of the IMB, the the Spanish National Bioinformatics Institute, and Sofia Fortler, a longtime colleague in the field of orthology and parallel, who has now her own lab in Berlin. ECCB also brought a number of discussions and so on, including a very interesting topic on mental health in academia. Today, we're starting ECCB 2020 with around 900 attendees registered for the main conference. And if we include the satellite event, we reach close to 1,000 people. We have 6 keynotes, 3 each day, together with parallel sessions to present 41 selected proceedings around 5 topics: data, genomes, genes, proteins, and systems. For the first time, ECCB will have a new session. And this session is about, or these 2 sessions are about global glimpses into bioinformatics communities, with Sol Bio as a representative for the Latin American colleagues and our colleagues from Elixir. There is a great opportunity to get to know a little bit better the development of those specific areas of the world. And there is a special for us to get closer to the Latin American bioinformatics colleagues in ECCB, using the virtual format of the conference. Today, we will also have the Connect and Network session. That is the name that we decided for the breakout rooms, where there are interesting opportunities to discuss specific topics in a more relaxed setting. Some of those sessions will be open to everyone and will show up in your timeline, so in the platform, while others will require you to register. Please register during this morning if you haven't done yet. From the logistic point of view, I would like to make some announcements. Sessions are strictly timed, so please keep to your assigned time for the good flow of the conference. We have the live Q&A to ask questions during each session, so chairs will be looking at them. We have enabled a Rocket Chat. You should have received an email on Saturday as a communication tool for the conference, so that will allow you to keep the conversation even beyond each of the sessions. You can always request help from us in the live support button in the top right, so you will see it in the top right of the panel. And we have a selection of booths from our kind sponsors and a career fair booth with some interesting opportunities. I would invite you to use the long break to visit them. And finally, last but not least, we have a code of conduct to ensure a pleasant experience to everyone during the conference. You will find more about that in our website and also in the every panel in the platform. ECCB 2020 have a special issue that will be published in the second half of October. So author will receive the proofreading by the end of September. Finally, I would like to thank everyone for collaborating and making this possible, including our ECO company, ECO Congress, who is supporting us across the whole event. Thank you.
Thank you, Salva. Now we start with the scientific session, and it's my task and my pleasure to introduce the first keynote speaker, Professor Modesto Orozco. Modesto is a career research professor working in one of the best institutes in Spain, certainly, and in Europe, at the Biomedical Research Institute, the IRB in Barcelona. There he has created a very large and extremely successful computational biology department. Modesto is a very well-known computational biologist specialized in computational biophysics. His research covers a wide range of areas and topics that go from the development of methods and for fields for molecular dynamics and for molecular simulations to relevant applications to biology, including protein function and protein dynamics, binding and design of small molecules and drugs, the structure of nucleic acids with a very interesting work in the formation of triple helix structures and the biological consequences, and more recently he has been working in the structure of chromatin with very large simulations. As a scientist, I admire Modesto's ability to combine the deep mathematical and physical understanding of the problems with the insight in the biological applications, including the experimental validation that is done in his own lab. At the personal level, I admire even more Modesto for his unstoppable capacity to stand against stupidity in science. Issue 1 my favorite anecdotes about Modesto, Modesto confronted a very high-level panel, review panel, composed of experimental biologists that were questioning his results and his approach, telling them that he was not going to enter in the detail of the explanation of the methods because they were not qualified to understand what he was doing at the mathematical and physical level. So that can give you an idea of his ability that I truly admire, not to let stupidity run science. With these few words, I welcome Modesto, and I really look forward to see what he has to tell us as the first keynote speaker of ECCB 2020. Please, Modesto.
Thank you, Alfonso, for the introduction and for inviting me. Can you see, Alfonso, my presentation? Can you see the presentation?
Yes.
Okay. So it's great to be here. It would be much better to be in Sitges, but if you want a snapshot of optimism, it's raining today in Barcelona. So it's not a beach day, so it's not so bad to be in remote. But you must go, you must come to Catalonia, and you must actually visit Sitges. It's really a wonderful, astonishing small town. So I'm gonna talk today about DNA, which is my favorite molecule, and I think it's a paradigm of the molecule in the middle, molecule in the frontier of physics, chemistry, and biology. So if you talk to a chemist about DNA, sure, he or she will say that DNA is his molecule, you know, is one especially complicated part, one chemical molecule. If you talk about biologists, he will probably say that, okay, DNA is the origin of life. And for me, DNA is a challenge. So it's a system that you need to tackle from the very small to the very big. And this generates, from a theoretical point of view, a problem of multi-scale physics. So when you work with DNA, sometimes you need to be worried about details that are very, very, very small. So, you know, a change in 0.3 angstroms in a hydrogen bond distance can ruin the interaction of transcription factor with DNA and actually give special phenotype. So we are talking about 10 to the minus 10, 10 to the minus 11. But another type of project, you are interested in this other level. So you are interested in working with anti-chromatin. For human, this is 2 meters. So you have to navigate in a 10 to the 10, at least into a 10 to the 10 scale. And this is a real multi-scale problem that needs to be faced with multi-physics approaches. And this is what we have done for many years now, trying to develop methodologies, technologies, methods, strategies, physics that allow us to move from the very small, that will be the kingdom of quantum chemistry, up to the very, very large, up to the chromatin level. And always trying to keep a kind of consistency, a kind of links between the different levels of resolution we want to do. So I'm going to make a very quick overview about how you can start from here and actually move up to the entire chromatin. I will show you results for it. Very small, I will show you results from the very large. And I hope to convince you that we have a kind of continuum in the methodologies that we are using. So let's go for the small. Small means when you're actually working with something where details matter, when you want to understand even where the electron distributions are. And of course, this is the realm of quantum mechanics. And it's not very common to see quantum mechanics in a computational biology conference, but I'm going to use 5 minutes to convince you, to try to convince you that it can be useful. And it can be useful in those cases where you don't have any idea, where you cannot compare, where things different to those that you know are happening. And I think this is a nice example. This is, I'm going to show you now, is the work of Juan that did the simulations and mostly that did the experiments. And you know, the guy I'm going to tell you about is a DNA scientist. So I'm sure when you think in DNA, you think in in a substrate, you know, the substrate of restriction enzyme or RNA polymerase, or as a product, the product of a ligase or a DNA polymerase. But DNA is also an enzyme. DNA can be also catalyzer, and this is something that many people don't really know. So you can build enzymes based on DNA, and they can be as efficient as those made by RNAs or proteins. And this is the only DNA enzyme that we know at the atomistic level. And this is a small piece of DNA you have here in gray, and it catalyzes the ligation. It catalyzes the ligation of two RNA segments. So red is RNA. So it takes this RNA, it recognizes it takes this RNA, it recognizes, and it makes a chemical bond. And this is a very difficult reaction. If you try to do by yourself in the lab, this is a pain. So we take the structure and then we try to understand how it works. And of course, we cannot use the standard bioinformatics method because we don't know it. We know how polymerases work, but this has no amino acids. It's a completely different chemistry in principle. So the first thing that we did, actually Juan did, was to take the structure and see whether it can be a catalyst. And the answer is no. The crystal structure that was published a couple of years ago is not catalytically active. And the reason for this is that there are no ions there. So the first thing that we did was try to build a catalytic active enzyme. For this, we use, basic Boson-Boltzmann physics, and very quickly, one found that there were two positions that were very likely to be occupied by magnesiums, even in the crystal structure of magnesium. So this is the configuration that after molecular dynamic simulations we found with the two magnesiums nicely coordinated. So it looks like a realistic, but before running the quantum part, remember, we want to analyze a reaction. In a reaction, you need to go quantum, so you cannot choose. Before running the simulations that are HPC type simulations, very expensive, what we did was try to see whether or not our model has predictive value. And something that was not obvious for an educated scientist was why there is a specificity for RNA and not for DNA, because the difference between DNA and RNA is the presence or absence of an hydroxyl here. And this hydroxyl is doing nothing in our model. So why you really want to have RNA, and if you put a hydroxyl here, if you remove the hydroxyl here, you lose the reaction. This is just to introduce you, this is the GDP, what is going to be transferred. This is the enzyme, a part of the enzyme, and this is where all the action is going to happen in this region. These are the two mechanisms. Okay, then very simple. We have an RNA, then we remove the 2' OS to generate the DNA, and then what we found is that immediately the active configuration is lost. So you see here, this is where this is the sugar packaging that is in the north conformation, it changes to the south conformation, and when you change, you lose the product, the linearity. So actually what it means is that you don't have a catalytic reactive configuration. Okay, so this is predictive power. So we didn't understand why, so we can predict something. So we then move to the quantum level. And for the quantum, of course, you cannot treat the entire protein quantum. We take a small part of the system quantum, the rest was classical. This is what is called PML. And I'm not going to go to the details of the reaction. This is one reaction coordinate. This is the other reaction coordinate.
Proton transfer.
And the other is the phosphate transfer. And you can see that this goes in this way. You can see here, this is the point more or less here where the phosphate is going to be transferred. You have here the PPI. You see here that the proton transfer starts to happen. So this is this downfield process here, and here you have the product. Then we can, of course, with this potential free energy surface, we can compute the activation free energy is around 24.5, which is really great. So we match the experimental number. This is the experimental number we also determine. But there is a bad news is that the reaction is not possible. So it's an exothermic, it's an endothermic reaction. So it's not 12 positive, doesn't make sense. So what is happening, of course, what is happening is that we are not recovering enzyme. So at the end of the day, you have here again the reaction. What you have is this situation where the DNA sign is protonated in this point, and this is not likely to be a happy end. So what you need to do, and this is a good lesson, you need to remember that you must recover the sign of the day at the end of the day, and then what we did was to see what can we do with this proton that is in the wrong place here. Then we can see that we can transfer this through a water molecule, we can transfer to this other position, to this uridine here, and from this uridine here again through a water molecule that is very well determined, very stable through the simulation, we can transfer to the PPI. And if you remember your biochemistry classes, PPI is a fantastic living group. This is why it's so good to degrade ATP. And when you have this PPI here, you have an exothermic reaction. So the proton is at the PPI, you have an exothermic reaction, but the very driving force is leaving the PPI. So this is this is a junior course in biochemistry, the energetics of the PPI, and it was a surprise when we move away the PPI from the site and we recover the site. It is downhill, a lot of energy is provided, and quite surprisingly, the two magnesiums at the end of the day, we recover the crystal structure. So the crystal structure was not wrong, it was just a silent form of the enzyme after the catalytic cycle was completed. Of course, all the model here depends on the presence of magnesiums. And remember, magnesiums were not in the crystals. We invented the magnesiums. What is the role of these magnesiums? This is these are kind of similar to what I showed you before. And you have here the reactant, you have the product, you have a nice reaction funnel for this reaction, to this pathway. And this is what happens when you remove one magnesium or the other magnesium. The enzyme is not catalytic anymore. So the DNA enzyme needs magnesiums. Of course, the problem is now that, come on, where these magnesiums came from. So we invented. This is what the reviewers of the paper tell us. Okay? All your mechanisms dependent on something that is not an experimentally validated fact. So we need to validate it, and this is what was the work of Mose. I'm going to go very quick on that. But the idea is very simple. It's a standard chemistry. Magnesium doesn't like a sulfur, so you have a phosphate and you substitute an oxygen by a sulfur, then you will expect a decrease in the catalytic power if this magnesium is involved in the catalysis, of course. And this is what you have here. You see a dramatic loss in velocity when you substitute the phosphate by diphosphate in this position or these other positions. The final proof, what is the final proof? The final proof is substitute magnesium by manganese. Manganese loves sulfate. So if you have as phosphate that you substitute by diphosphate and decrease the velocity, and you add manganese and you recover velocity, this is 100% sure proof that this phosphate is involved in a magnesium interaction that is crucial for catalysis. And this is what we find here. So these are numbers relative, these are control. These are numbers relative to the situation with magnesium. You see that you have 2 orders of magnitude, this 1 or 2 orders of magnitude effect that is actually confirming that these are magnesiums. We double check it by this order reaction. It shows that there are 2 magnesiums there, and we verify the proton transfer to kinetic isotopic effects, and again we validate them. So I think this is just a snapshot, an example of how nice chemistry, nice biology you can do using quantum chemistry in those cases that you are facing unknown territories. So in quantum chemistry, you don't need to know anything. You just follow first principles, and then you have a technique that is universal in use. The problem, of course, is that you just can have a small system or a small part of the system. If you wish to study big systems, you need to go classical. And this is the kingdom of classical mechanics. As Alfonso said, we have been working in fossil for many years. This is part of the C1 that is actually the gold standard in the field. It was the work of Ivani, and many other students and postdocs in the group. It was more than 6 years of work really to calibrate the function and make it trustable, and making sure that the function has predictive power. I think it's the first function with predictive power. I'm showing here an example. This is St. Thomas, and I put this slide because of some referee comments of whether you can really predict instead of just reproduce what you already know. And this is the work of Pablo, and this is a kind of proof of concept kind of thing. So the idea was, okay, they ask, or they tell us, or they criticize us that we don't have predictive power because we are reproducing what we already know. So let's go and take two sequences that are not in PDB, so we cannot guess how they look like. Are actually quite strange. If you see these ones are not useful structures. And then with the help of Carlos Gonzalez, we solve the structure in solution by NMR, and then we try independently, completely independent, try to get the structure from the beginning, from molecular dynamics. In this process, we realize that actually NMR is not, it is quite, it can provide quite different results depending on very subtle details. So we decided to go a kind of blind test. This is a kind of control. And this is really because of the Decamer is a duplex that has been solved 20 times. And there is one structure that is, everybody agrees, that is a gold standard, that was obtained using all kinds of restraints and data on solution. Okay, this is the control. This is the gold standard structure, 1 NAG. This is our NMR structure that we solved from scratch. So we use the standard procedure that is using 99.9% of studies of NMR, and this is our a priori structure for molecular dynamics. So, of course, the structure looks exactly the same. You see RMSDs that are ridiculous. We are closer to the NMR
than the crystal.
or other alternative older structures. But if you go to the details, this is very interesting. So you see many, these are tweaks, that is a kind of helical parameters that give you ideas about the local structure of the duplex. And you see that there are very many which are very similar, and one here in green that is completely different. And you immediately will say, okay, this is the MD. No, it's not. This is our NMR structure. So we did our best, and this is what we get. So when you then take a look to the observables, you play with the refined model, I don't have time to go to the details, but actually you do the refinement better that is typically done, then you recover this NMR start structure that is actually very, very close to our trajectories, but very, very close, almost indistinguishable. So the take home message is that, if you take care if you don't take care with NMR, you can have results that are a little bit tricky. If you take care, you can really have very good results, but in any case, are not better than them. So then we do the real test, sec 2, sec 3, and this is what we got. So these are results. These are structures that the NMR and the experimental NMR and the MD simulations are indistinguishable. This is a part of the business. So we are able to predict structures that are stable, and we are able to reproduce the structures. But what happened with those that are not stable? And these are very recent results from Monse that did the experiments, and Vito that did the simulations. And we study here a bunch of triplexes made with RNA and DNA, and this is because we are studying the regulatory mechanism based on hybrid triplexes. It was a kind of control test, but the results were surprisingly nice, because we found that after not too large simulation, these two triplexes actually start to unfold. While this order remains folded, I would say right now for more than 1 microsecond. Do the experiment, and then this is what you find. In these cases, Monse found melting temperatures very close to 0 Celsius. In this case, melting temperatures were in the order of 40 to 60. So what we are finding here is, again, predictive power. We are able to distinguish what is unfolded, what is unstable, to what is stable. We have used BSC1 for many, many studies, and I'm going to just make 2 snapshots to very quick insights to demonstrate or to convince you that they have predictive power in the multi-sig. First is the wall of Fede.
With colibactin.
Colibactin is a toxin generated by E. coli, and our colleagues in Germany and the US demonstrate that it's actually very tightly coupled with colorectal cancer. The challenge here is to understand why, and then the other simulations strongly suggest that colibactin is acting as a metabolite binder, and here it has this cyclopropane that actually binds in here covalently, binds in here covalently, and then very later on there is a break in the beat. What was the real shocking result here? What was the sequence preference? So why the break happens in things that are rich in this strand sequence, AATT or AAT. And this is what MD tells you. What you have here is a bunch of different structures, and you compute different physical parameters describing the DNA. For example, this is a stiffness, how rigid it is. You know, this is the major group wide. So what is the width of the major group of the DNA, of the minor? So, and then you can see in the middle that there are two sequences, two tetrameric sequences, which are completely different. And those are that are found experimentally generating the lesions that produce the cancer. So predictive power, again, this is the work of Antonio. And again, I think we showed you the predictive power. Here, that it was a kind of a side product of the parameterization. The objective was see whether or not the fossil was able to reproduce the crystal. Not the crystal structure, we knew that we reproduce well crystal structure. We want to see who could reproduce the crystal. And then we took Druidicus of the decamer. It's found in the PDB in 3 different crystal lattice. And then we set up as close as possible simulation conditions for reproducing the crystal, recreate the crystal. So these are systems which are huge. So I'm talking again on an HPC problem. The system goes beyond 1 million atoms. And then we compute the RMS differences between the strands. So within the strand, everything looks okay. But if you look at the distance, at the movements of the helix, one in front of the other, we have many helices in the simulation cell, then you see that in the plane, there are no big differences, so the helix doesn't move too much. But if you look at the z-axis, there is a dramatic difference. So what it means is that in this simulation system, and in this simulation system, there is a slide. The crystal is lost. And this is a surprise. It looks like the fossil is wrong. But then it's surprising when you see that in this other crystal lattice, everything looks perfect. And this is inconsistent with the first scenario. So then we take a look at what happened in these two others and why these two others were different to that one. And looking in the paper, we realize that the only difference is that these crystals, these two crystal structures that you see that they unfold very quickly, were obtained in the presence of spermine. So the additives that you add in the crystal may have an importance. What we did then was to repeat our simulations, but increasing the amount of spermine in the crystal, and actually you see that you converge to the crystal. So the take-home message is that you don't have to, or please don't forget the crystal additives, they are important, but there is a more important biological consequence, is think about these unstructured cationic peptides when you think about chromatin compaction, because they can actually generate a specific phase that can stabilize chromatin when you don't expect that this chromatin aggregate should be stable. This is actually what you find here. Why you don't see this spermine in the crystal? Very easy, because they are moving very quickly. They are not placed in any fixed position. They are generating a kind of plasma that stabilizes the aggregation of the chromatin. Okay, this was my second step. Let's go to the last step. What can we do to study big pieces of DNA? And when I say big, and we are studying now human fibroblasts, we have characterized well yeast chromatin. What can we do? Try to keep physics as good as possible. And this is our approach. This is the work of Julian Walter and actually many other people in the group. And it starts from a molecular dynamics simulation. From molecular dynamics simulations, you realize that, you know, very often you find this type of distributions that are Gaussian-like when you project into the into the helical space. But somehow what you say, you assume, is that instead of looking at all the Cartesian degrees of freedom, you can transform in terms of 3 translation and 3 rotation degrees of freedom. And as they have this Gaussian shape, you can define a kind of a stiffness matrix. As you can see in many of some cases, there is not one Gaussian, there are several Gaussians. Then we play with statistical mechanics and develop a method that is actually related to empirical valence bonds or Warshaw's ideas. At the end of the day, it means that you can reproduce by means of a series of stiffness matrices that have formability. And these stiffness matrices are very, very useful because they tell you what are the physical properties of the unit. And you can use this for many different things. For example, this is the work of Alba and Fede. We can use this stiffness matrix and equilibrium values to describe the binding of DNA to transcription factors. The relationship is not obvious, so we need to use machine learning. In this case, it was a random forest regressor, similar results with other methods. But the idea is very simple. So you take selects data, that is your experimental input, as your parameters at these stiffness matrices. So really no sequence here, just properties, physical properties. And this is all the data I'm going to show you is in the validation set, and you can see the correlation. And well, of course, you may think, okay, this is one case. Here we put all the proteins for which we have data. And you can see that there are some exceptions, and I can talk you about what happens here. But you can see that of the 70 protein, in most cases, we have a predictive power above 0.8 in terms of correlation coefficients in the test case. So these parameters derived from MD simulations have real predictive power. They can predict biology, and they can predict even things that are more complicated. And I'm going to move now to the chromatin field, particularly starting with the nucleosome position. This is the standard MNA-sec experiment. We use, in most cases, we use this. You treat this with MNAs, that is a nuclease that degrade the DNA if it is not protected by a nucleosome. And at the end of the day, what you have is that only the segments that are covered by nucleosomes remain after the degradation, and you sequence them, and then you map. A few years ago, we proved that actually there was this kind of profile, that this will be the promoter region here. This will be the +1 nucleosome, the first nucleosome in the gene. This will be the -1 nucleosome, and then we proved that actually this nucleosome region in the beginning of the gene is marked usual physical properties and by the presence of transcription factors by the site. So we can take then these two signals, the blue and the green one, and train another, in this case, it's an extra random forest, another machine learning. This is the rock cube you get. You can predict many, many nucleosome regions by these two observables. So known bindings of transcription factors and regions with unusual physical properties. When you have that, you have the nucleosome region here, and you have another one at the end of the gene, and you can use signal transduction, signal transmission theory. This is the work of Diana and Paul. Both are mathematicians, and actually they play with this idea that the nucleosomes are positioned based on two exclusion signals, one at the beginning of the gene and another at the end of the gene. These are like two emitters that send signals of positioning, of periodic positioning from different sides, and this will give you two kinds of localization of the nucleosomes, those that are fuzzy and those that are well positioned, that are the majority. So this is experimental data. It means here these are all the genes in this. The data was collected by all the engineers in the lab. This is the +1, +2, blah, blah, blah. And then you can see how there are cases where are fuzzy, and there are cases where are very, very well positioned. It is easy to demonstrate that both cases can be explained by this kind of signal decay. And for me, what is more exciting is that we are able to predict well the entire chromatin structure in terms of the nucleosome based on principles that can be derived directly from the dissimulation after manipulation of the trajectory. But we can go further. So we have now this method that allows you to compute, let me move here, you have this stiffness matrix, and using very simple math, you can compute the energy associated with this stiffness matrix, and what you then did was to add the nucleosomes, because we want to study chromatin, so we need to add nucleosomes. These are the type of movements we built up. We sample through metropolis. This is the way in which we treat the nucleosomes, very simple, so the vehicle plus energens. And with this method, we can actually start trying to make sense of MNA-sec data. And this is an example of how we publish our results in terms of nucleosome position, and this is a real truth. And the real truth is not as nice. So you know, it's very likely to believe you have a nucleosome here, but it is, you know, and here is a binary. So we put 3 nucleosomes, but come on, you know, and what is happening? What is happening is that really what you are finding is a population. And in this population, you have places where all the cells have a nucleosome here, and you have others in which some cell will have a nucleosome here and another here, other population of cells will have one nucleosome here. It will be a little bit fuzzy. So you have the population problem, and this is the way in which we solve it. This is just a detail of how we parameterize the nucleosomes of the vehicle. This is how we feed this probabilities to reproduce sedimentation. But let me go back to this map. When you have this map, the most annoying thing that you have is the noise. And the noise is killing you, for example, when you like to, or when you want to compare 2 different experiments. So let's say that you have a cell, and then you determine the nucleosome map, and then you deduce what regions are accessible, which are not, and then you add another one or do something, and then you want to see what are the changes in the nucleosome. This is completely corrupted by noise. So Diana and Oscar and many others in the group designed this method that is available, so you're free to use it. It was published a few months ago. That allows you to compare directly the restriction maps, so the degradation maps, and actually determine what are the differences in the nucleosome architecture when you introduce changes. And this is an example. This is from our own work. This is a cell cycle. And I put you here, this is global, I don't have time, but I put you here a few examples. So this gene is expressing G1. This is expressing S, and this is expressing S. And this is a nucleosome organization in G1 and S. And this is the type of changes that we are finding. For example, this is an addition. And you can see it. I'm sure that all of you will recognize where the gene is active just by looking to the changes in the nucleosome. Here you have, for example, the promoter collapsed by this nucleosome. This gene is going to be silent. When you edit, and you have here the signal, you edit this nucleosome, actually is when the gene becomes active. This is another example in diet. This is a normal yeast that is fed with glucose. Then you remove glucose and you add galactose. In this case, what is happening is that all the enzymes that transform galactose into glucose are fired, and you can see here there are very little questions that you are actually fighting the enzyme. You see that this transcription factor binding, this transcription factor region, this promoter region was collapsed. This is fired, this is fired, this is fired. Here is just dramatic, you know, the effect. That is a silent gene, and here you find this dramatic division. Actually, it goes active. And here even more dramatic when you remove glucose, you remove galactose, and you add ethanol. Yeast doesn't like ethanol, and then all the heat shock system is fired, and this is the type of response you have. You see these dramatic changes in the nucleosomes that are traced. You see here, for example, all the massive depletion of nucleosome in genes that need to be quickly activated. With this technology, we can face a very difficult problem. How can we understand this type of plots? This type of plots, I mean, you can put a nucleosome here. I'm sure you can put another here, another here, another here. But here is science fiction. And know that I'm criticizing my results. But this is how life is. What is this map of RNA-seq showing you is that actually we have many families. And what Julian, Diana, Pablo, and many others, Juan Pablo and many others developed is a method to deconvolute the signal into a series of families. So this is, for example, in this case, with 5 families, we can reproduce the experimental RNA-seq, and we can put structures to these families. So how can we verify that this is right? Of course, the physics of this is properly determined by the method I told you, but we have experimental data, to put extra experimental data, that in this case are micro-C data. For example, we know that this nucleosome is close to this other nucleosome, and these are the strains that we are putting to bias our simulation. So at the end of the day, we have a series of configurations which are compatible with the physics of the system, and that can explain all the observables that we have. And the results that you get, or you can get, are really exciting. For example, this is our own data. This is yeast in normal conditions, and this is under oxidative stress. When you see the data, well, it's not dramatic differences, but when you put the 3D structure, you realize that this is a much more extended structure, and this is much more convex. Very interesting, this is a gene that is activated as response to oxidative stress. In wild-type conditions, it is completely repressed and is actually, partly dense. No way that it can be recognized by the polymerase system or the transcription factor. When it has to be active, you see that there is a dramatic extension of the gene.
Modesto, we have 3 minutes if you want to have questions.
Okay. Okay, we are moving faster to the chromatin fiber. Here you need to put, the first thing is to put is average strain by high C, but we put many others like telomere publication, microscopy data, ideas about the volume, the shape, everything that is around. And I don't have time to go to the details, but this is the type of thing. These are data from humans. This is fibroblasts. These are 2 genes we are interested in, a different arrangement of the chromatin. You can see that we got ensembles, so many different arrangements. And what is important here is that we go to the base pair resolution level. So if you really wish to understand biology, you need to go to the base pair level, because this is the level where precision factor access. So you can see here, this is more detail, we can map it. These are nucleosomes, and these are base pair. Okay, I have here, how much time do we have, Alfonso? Okay.
So we have 3 minutes to finish the talk and then 10 minutes for questions, but you can take some time.
Okay, so this is an example of how far can we go from the beginning to the end. This is a study on the effect of DNA methylation. And it starts from molecular dynamics, where you have this is a CpG step, this is a control one, and this is the same step here but methylated. And this is a thing. When you see here things that are very red, means that this is a very stiff distribution, that is a very stiff step. Look here. When you see this kind of distribution, it means that it's very, very shallow, so this is very flexible. And what we found here is that when you methylate the CpG step, it becomes very stiff. This is expected to have an impact in nucleosome organization, and this is what we found. This is again RNA simulations. We methylated the DNA, in the major, in the minor, in all cases it's positive. So it is the effect of the methylation of DNA in nucleosome binding is to make it much more difficult. And this is experimental validation. So I'm not talking only about simulation, these are the experiments. So the take-home message is that when you methylate DNA, nucleosomes are unhappy. And this will suggest that this will make an effect on nucleosome organization, a kind of chromatin remodeling, that this can be actually coupled with a difference in function, in the different activities in the genes. And of course, this was just in vitro. Then we decided to do studies with cell system. Our cell system was yeast. We take this, we methylate it. So we add methylases. Well, this is a way in which you can actually create a new organism that is adjust with the properties of a mammal chromatin. At the end of the day, these cells are stable, and then we look at where we validate that actually this DNA was methylated, and we predict where the methylation happens. And in principle, as this has no methylation machinery, has no way to recognize where the methylated DNA is, no MBD, no TET, nothing, you should expect a random distribution, and actually is not a random distribution. So you have a dramatic decay near the transcription start site, and then you have an increase in the methylation level. This is like in mammal. We predict that methylation never happens in nucleosomes, and this is indeed what will happen. These are regions with nucleosome. You see, regions without nucleosome is random. Regions with nucleosome you have in the linkers. There is no methylation in the nucleosomes. Does the methylation has an impact in nucleosome organization? You can see that it does a dramatic effect. Actually, it moves the transcription factor, it changes the way in which the DNA is recognized, it makes smoother the path, and it has, a dramatic effect in the global organization of the nucleosomes. That actually is very easy to see that couples with the activity. But it also has an impact in the global chromatin structure. For example, you lose a lot of long-range contact. You gain many short-range contact, but you lose many long-range contact, and actually give a completely different path in the chromatin. So, for example, chromosome 12, this is our reconstruction, this is the ensemble we have, and you see that this is a dramatic loss in long-range contact between the chromosomes. And in some cases, this can have, this, for example, is a region that is important in division, and you can see that there is a dramatic change in the distance between the two genes that actually couple with the phenotype of the cell. And what is more important for this, remember, this has not any methylation recognition. So the only explanation that all this happened, all these changes in chromatin, all the changes in function of the genome, is that it goes through a direct effect, as we suggest. The direct effect can be that the binding of transcription factor is different because there is this methyl group there, or can be, as we believe, because the stiffness of the DNA changes, nucleosomes are rearranged in a different way, and this impacts into the function of it. I'll finish here. If you wish more details in this last project, I can explain you. This is my last slide, just to show that what I have put here is just in a very, very small part of my own work, is the result of the work of many people. So thanks them for the work, and thank you for your attention.
Thank you, Modesto. Pretty impressive, as always. Before we get the questions from the chatbot, let me ask you something that is going back to the beginning of your talk. You have made a very clear exposition of the modeling that can be done at different levels of resolution, from the atomistic level to the chromatin and the nucleosomes. And you explained at the beginning the importance of multi-scale modeling, but still, if I would say that these are still modeling that at different levels, but it's not an integrated approach of modeling at different scales. Do you know what I mean?
Yeah, you're right. For example, so for example, when I'm very excited about things like diffusion and factors and binding. So transcription factor is, can be 3 nanometers. And when you play with high-speed data or microscopy, you are typically in the micrometer range. So to really go to learn chemistry, you need to go to the base pair level. But of course, we cannot build yet a base pair level for the entire chromatin. So what we can do, and this is correct, is that we can rebuild pieces of the chromatin at the base pair level. Right now is a computational issue. It's still not possible to build the entire chromatin simultaneously at the base pair level. But yes, I mean, in the way in which we work, we have base pair resolution at the mesoscopic level with the constraints that are given by the fiber that in the case of micro-C is 200 base pair resolution, in the case of high C is 1000 base pair resolution, microscopy depends. So the coarse grain level is integrated in the lower level, but of course we need to break into windows. We can integrate atomistic with a coarse grain with a mesoscopic, and we have done. So it's a big piece that is treated at the mesoscopic level with a part that is treated at the molecular dynamics, but we haven't done integrate everything in a single frame. This is very, very complicated, but it's mostly a computational issue.
Thank you, Modesto. We have some questions. We have only 3 minutes, so we better go directly to the question. The first one is you think that the presence and the concentration of magnesium affect the effective recognition and the binding of the proteins, specifically the transcription factors?
This is a very good point. I don't know. So probably in some cases, for example, if you are thinking in catalysis, magnesium is extremely important. In no catalysis, I mean, we have done a survey of PDB, standard bioinformatics study. This is the work of Diego. And in many cases, it's not clear that PDB was labeled as magnesium with the magnesium. So in many cases can be solid. So my guess is that magnesium is absolutely crucial for catalysis. In terms of structure, is perhaps less important than what it was thought. It's clear that magnesium, has a very important effect in and so on. But yeah, so in many cases, the job of magnesium can be done by other areas.
The last question in 1 minute is what can be expected in the study of protein and DNA interactions at the other 2 levels, the first level, the quantum level, and the second level, the double helix type of structure that you were talking about in the second part of the talk?
I think that for the biggest thing that has been done to my knowledge is simulations that we have done, but still just proof of concept that are entire genes. So we can take a gene from yeast and simulate it at the atomistic level. But these are proof of concept. I think that and from the quantum level is the things that I have shown you. So for the rest, I believe that we need to go to this composite level. So take a part of the high resolution level and the rest of the low resolution level. Okay.
Well, thanks, Modesto. I think we have to, the system works automatically, so they will kick us out in 30 seconds. So I use the opportunity to thank you for the talk and for the explanations of the different levels of modeling and how to integrate that with beautiful examples that goes from the enzymatic capacity to the long-range interactions in the chromatin structure and the nucleosomes. Thank you very much, and we continue with the next session.